Our next speaker this morning is Dr. Sylvia Vetrone, who is an associate professor of biology at Whittier College, a private four-year undergraduate institution in Whittier, California. She earned her PhD in molecular cellular integrative physiology from UCLA and joined the faculty at Whittier in 2008. Professor Vetrone's cross-disciplinary interests in immunology and oncology guide the various research projects that she conducts with her students. Among those are one, understanding the role of plant nutraceuticals on lifespan, oxidative stress, and antimicrobial responses. Secondly, the validation of DNA-based biosensors for the detection of foodborne pathogens. Thirdly, the role of cell migration in cancer metastases. And fourthly, T-cell mediated immune responses in disease. Today, Professor Vetrone will describe her work in nanoparticle-based sensors for pathogen detection. Professor Vetrone, welcome.
Hello, thank you, and thank you so much for the invitation to come and talk to you about a collaborative project that I became part of back in 2010. So as mentioned, I want to give you a little bit of background of how I jumped into the foray of nanoparticles. So as mentioned, I'm at a primarily undergraduate institution. We are nowhere near a big R01 institution like here in Minnesota or at UCLA. And so one summer through the Department of Homeland Security, I applied with some of my students to go to Michigan State University to engage in a new collaborative effort. And it turned out that the PI there, Vanji Elosiha, is a biochemical engineer, and she herself works in creating nanoparticles and also various biosensing strategies for detection of many different analytes. And when I came to her lab, as a person who's not in the field, again, I'm an immunologist, I really had questions about, okay, these are working in the laboratory setting, this is fantastic, but how do they actually work when you get into the field, right? So can you take it from a very expensive, labor-intensive setting to a more simplistic model out in the field where maybe even individuals that don't have the economic resources can actually use them? That was my first question. My second question is, what is the toxicity of the end effect of using these nanoparticles once they get out into the marketplace? What is the end effect of having all of these in our environment? So today I'm really just going to talk about the first part of it, which is testing the biosensors using nanoparticles to their limit to see how we can essentially use them when we expose them to different types of matrices for those analytes, and then streamlining that process of detection, because that's also really important. So we've heard a lot, so this is going to kind of speed up my talk. And again, this is kind of nice because I'm right before lunch, so bear with me. All right. So again, why nano-based biosensors for detection? Specifically gold. We already learned a lot of the properties of gold, but I'll go over that again real quickly. Then I'll really get into the DNA-based biosensor that I'm specifically been working with and how I went about validating them. And then what are some things to think about as we think about streamlining that process? So first and foremost, today we heard a little bit more about therapeutic applications of nanoparticles, but the reality is that nanoparticles are being employed for surveillance efforts, whether that's surveillance of disease or surveillance of would-be biological threats due to public health, threats. So when we think about surveillance, we think about three things, which is essentially prevention, protection, and mitigation, right? So we want to make sure that there are no public health issues, right? We want to prevent them. We want to also protect ourselves against them. And if they are occurring, how can we quickly know that one is occurring and then put in protective measures after that? And so here's just an example of various, of various different industries that use nanoparticles for surveillance. So again, water safety, food manufacturing and distribution. A lot of that surveillance happens either on-site or off-site, or in the line of getting it from where it's being manufactured to where it actually ends up. Agricultural, we know that recently there's been a problem with lettuce, right? So we also hear about recalls often, a lot of salmonella, a lot of E. coli contagions happening in our food supply. Environmental monitoring is very important. Of course, we heard a little bit about medical diagnostics here today, but they're also used in infection control. Again, testing laboratories. A lot of these individuals, whether they're in the food or environmental monitoring, they actually don't do them, but they send out samples to get tested at various test laboratories. And then, of course, military biodefense. So a lot, Department of Homeland Security, Department of Energy, right? A lot of them use these types of surveillance systems. So the way in which they do this is that they use very conventional methods for the most part. And these conventional methods reach out to detect various analytes such as proteins, chemical molecules, maybe some energy byproducts, right? And of course, various oligonucleotides, whether they're RNA or DNA. And the way in which they sense this is by using conventional molecular-based methods, such as polymerase chain reaction, whether that's just a general for a yes or no, or more sensitive, such as qPCR. But when we're talking about pathogens, especially on the level of bacteria, a lot of it is just basic culturing methods that, and then of course there's hybridization, but there's also those that use antibody-based methods such as Western blots, ELISAs, agglutination, and also flow cytometry. So the issue with all these methods is even though they're very sensitive and they're very accurate, some of them can create cross-reactivity where it gives some false positives or false negatives. And all of them, while again they're highly sensitive, are very time-consuming. So it requires a lot of time. It requires the use of a lot of equipment, instrumentation, and because of the equipment and instrumentation, that also requires skilled labor, so skilled personnel. And of course, all of that put together becomes very expensive. So this is a very rate-limiting access point for the need of other individuals or other agencies that do not have the economic means to do this. So income, nano-based biosensors. So these, what's really nice about them is that they're quick, they're reliable, and they're very relatively inexpensive. Similar to the convention methods, they're specific, they're sensitive, they actually have less cross-reactivity than those that are mediated by antibody use. And like anything in when we talk about specificity and sensitivity, we want reproducible results. So they're really good at providing that. The other thing that's really nice is that unlike conventional methods that can take anywhere from 1 day to even 10 days to get your end result, with nano-based biosensors, you can get a very quick response. So I'm sure all of you at some point have done some airline travel, right? And you get pulled off to the side and they swab you a little bit, and then they put that right into the machine, and then within less than a minute it gives them a result. That's a yes or no result to that analyte. So it's very quick to that endpoint. The other thing about them is that the reagents and even the instrumentation that is being used has a really nice shelf life. So when we think about PCR, conventional methods, PCR, culturing, ELISAs, that all requires that the reagents have to be stabilized at either freezing temperatures or even just refrigeration, and that can then also impact its ability of use. So the nanoparticles help to stabilize and extend the lifespan of the various components of the biosensor. And then all put together, they're very quick and simple to use. So again, I use that example of the airport, right? They just swab your hand and put it into the machine. So it's very easy to use, so easy to train. So when we think about this then, there are many different platforms. We've heard about those platforms today, right? And here are just some various lists of different types of nano-based particles that are used in biosensors. So we've talked and heard a lot actually about gold today. Some silver nanoparticles were introduced in the previous talks. And the other thing that we also saw, I think, were some nanotubes in some of those. So you put nano in front of something and you can create anything, right? Chips, arrays, tubes, nanowires, right? So being able to create these nanostructures, we've gotten really good at that, right? And so again, what's really nice about these nanoparticles is that they all have different properties that allow for detection. But when we think about all of these platforms, at the end of the day, they all, when put into a biosensor type of situation, they all have something in common, which is they all have these 4 different elements. So at the very beginning, they're all against, and they're all created to target a specific analyte. So that analyte can be organic or inorganic. And the thing is that the biosensor also has to have a bioreceptor, so something that's going to be able to latch on to that analyte of interest. So it could be anywhere from an oligonucleotide, antibodies, enzymes, other proteins, aptamers, even phages against various bacteria. But once that biosensor has latched on to the analyte, you need to be able to somehow transduce that to a measurable signal. And that then comes into what is that signal going to be, right? So it's going to be either optical, electrochemical, or even mechanical if it's like a cantilever system. And then you need to be able to capture that signal to measure it. And so the output system then is going to be your one piece of instrumentation, if you will, right, that's going to capture the luminescence or the fluorescence of the system. Maybe it's absorbance, so you're in that UV vis, or some magnetic resonance. So this is why gold nanoparticles are of a strong interest, and a lot of the literature, a lot of the research efforts are really using gold nanoparticles. So there are 4 main benefits to them. The first one is that they're highly compatible, to so many different organic, metallic, and biologically active molecules. Because of that, and again, we learned today from our 2 previous talks, right, that because of their small size, they have a very relatively high surface area to volume. So you can really then load them or cover them to serve as really effective carriers. Then, of course, they have a high attraction value because of their surface, right? We've learned today that we can essentially manipulate them or coat them to have either different charge properties or be able to effectually functionalize and add various bioreceptors onto their surface. And so then they can carry that forward. And because of that, the nanoparticles themselves, so the gold themselves, and or what you attach to the surface of the gold nanoparticles, create a very nice detection platform to the point of it being spectroscopic, colorimetric, fluorimetric, or electrochemical. So taken from these 2 review articles, I just wanted to kind of show you a little bit about what that may look like. So we've seen various representations of this, of the gold nanoparticle being functionalized and stabilized, right, by various bioreceptors on its cell surface to carry what it is we want to a specific location if you're doing targeted therapy, or, right, to serve as that bioreceptor capacity. And so depending on the way in which you're employing it, right, you can have different types of output. So in a colorimetric fashion, the thing about gold nanoparticles is that when they're loose and they're nicely dispersed, they can have, depending on their size, a very different variations of a red color. And the larger they get, or the more aggregated they become, that changes the spectrum of the color that we can visually see, and it's more in the dark blue, dark purple kind of range. So gold nanoparticles can then be used, right? If they're binding to your analyte of choice, that allows them to aggregate, which changes their size, which then physically changes the color that we see. So this allows us to be able to use absorption, if you will, and outputs to see if our analyte is there. Electrochemical, what this allows us to do is that depending on your system, so here in this cartoon, we see that if the gold nanoparticle is able to recognize and detect the analyte of choice, that's going to change the overall shape of the gold nanoparticle, making it heavier, and that then is going to impact how the current is generated when you hit it with some voltammetry. Last, I guess almost, right? Spectroscopic, this changes, again, the wavelength that can be captured, okay, depending on whether it's bound to analyte or not. And then fluorimetric, this is more when we think about competitive types of assays, where if your analyte of choice binds to the bioreceptor, that's going to bind to a higher affinity, releasing the fluorophore, which then is dispersed in the liquid, and then it's going to be giving you a higher intensity. So in this type of event, the more analyte you have, so the more target you have, the more fluorescence you're going to get. Okay? So specifically in the gold DNA-based biosensor that I've been working with, our schematic is to essentially use it to create a daisy chain or a sandwich-like structure. So our analyte, right, our target of interest is actually genomic DNA from pathogenic bacteria. So specifically, I'm interested in using biosensors for mitigating bioterrorism, right? So bioterrorism that happens on purpose, or even just in food industry, right? So Chipotle, again, the salmonella outbreak that's currently happening in the lettuce. And so what this does then is that both the gold nanoparticles and magnetic nanoparticles, which you can actually just purchase, is nice. So again, similarly, right, we're going to functionalize their surface. Here, what we have is the bioreceptor that we put on them are thiolate conjugated, and they are essentially just primers to a specific gene element in the pathogenic bacteria. So the bacteria of interest that I've been working with is Salmonella enteriditis. And so these primers are against the IEL element that is only found in this particular species of Salmonella. And so if the Salmonella is in the sample, what that's going to do is it's going to be able to bind to or be sandwiched between both the gold and the magnetic nanoparticle. So what's really important about this is that if we were to just have a system where we have our sample and we put the gold in there, it's going to be able to latch on if our target of interest is there. But how are we going to be able to pull that out? And so this is why we're employing the use of magnetic nanoparticles here, so that the only way you would ever be able to pull out the gold, which is going to be our output, is if it has been essentially tethered through having our analyte there. And so again, you can pull it out. Everything that didn't bind is going to essentially create no signal. So once we're able to pull it out, then we can just do some differential pulse voltammetry. Please don't ask me how exactly that works, but we essentially hit it with a voltage. And what that lets us do is that gold gives a very specific peak, and it's usually somewhere between 2 and 4 of 0.2 and 0.4 of voltage. And so what that, in our particular system, the more gold, the higher the peak. And the more gold there is, that would go to suggest that there is a lot more of our target there, right? And so what this system allows us to do, it allows us to do two things. One, it gives us a yes or no, right? If you're getting gold, then you for sure have your target in your system. But depending on how high that peak is, that also lets us know semi-quantitatively how much of our analyte is in the system. So that is, in theory, how it works and how we were able to show this. So to begin, this was done by a postdoc in Vanji's lab. And essentially, what they always show is that it always works in the lab. So just like any disease, so again, as an immunologist, right, we can give any drug to a mouse or do anything to a mouse and we can cure it. We can give them a disease and then we can cure them of the disease. But we know that that always doesn't translate to the real world. It's the same thing when researchers are creating these biosensors. They're always testing them against very purified, amplified target. So because you want to know if it actually works. But we know that in the real world, we're not going to have a nice, pure target, right? So we need to make that messy to see that if in our sample, the target isn't just going to be like, here I am, there's a lot of me, right? It's going to be in a milieu of many other things. So the biosensor needs to be able to essentially target that out, bind to it, and essentially ignore everything else. We want it to be highly specific. So the way in which I began to look at this then to validate it is we have our pure culture of Salmonella. We grow that up. This is just an overnight culture. And then what happens is that you isolate its DNA, and then you apply that to the system. What is conventionally being used right now with a lot of biosensors is that once they isolate the DNA, they amplify it. So they amplify that target using polymerase chain reaction. And as I told you before, that requires skilled labor, right? And I use air quotes with that because we know that you can really teach anyone to do PCR, but to then really be able to understand it is the next tricky point. So it's still skilled labor, and it's very expensive, and it's time consuming. So once that they've amplified that target, then they apply it to their biosensor system. But I wanted to step away from that. I wanted to know, does this biosensor work with non-amplified DNA target? So just naked genomic DNA. So I skipped the PCR, right? So I just grow up the Salmonella, isolate its genomic DNA, and then apply it to the biosensing system. So first allow it to hybridize to the magnetic nanoparticles, and then bring in the gold nanoparticles, pull it out, apply it to a screen-printed carbon electrode, add a little bit of hydrochloric acid to disassociate the gold from everything else, and then apply some differential pulse photometry to look at the peak. So as I had mentioned, right, when you look at PCR amplified DNA target, so this is very, very pure, a lot of target in the sample, of course this is going to work. And we can see that we can get down to the sensitivity of about 100 nanograms per milliliter, which is well within the range of specificity detection of a PCR method, right? And that makes sense because this is PCR amplified DNA target, right? But when I wanted to know, is this going to work with genomic DNA, so non-PCR amplified? And so I used Trizol, which is a very commonly available reagent to be able to extract genomic DNA. It works pretty quickly. It takes about an hour and a half to get your genomic DNA. And then I essentially apply that to the biosensor system. And once again, I got very similar results with the sensitivity down to 100 nanograms per milliliter. So similar to what we had seen with PCR amplified targets. So this was very exciting for us. The other thing about this, though, that I do want to point out is that you can see that the more DNA target you have, the signal goes down, right? And so this is something that was occurring often in our detection system. And so I'll talk a little bit about this later, but what was happening is essentially what happens in a lot of sandwich-like systems, so like a Western blot or an ELISA, is that when you have too much of everything, things start to aggregate, and you lose your signal, basically. And that's called a hook effect, and that's essentially what was happening in our system. But the one really nice thing about this is that you don't need that much DNA in order for this biosensor to work, which is even better. So the next step is, again, thinking about what about a laboratory that doesn't have a lot of money, or they don't have access to buy Trizol, right? Are there other ways that you can isolate DNA? And of course there are. There's the good old standard phenylchloroform method, which uses reagents that you can buy at a fraction of the cost of actually getting the Trizol reagent, and it allows you to do more isolations as well. So this is actually very cost effective. And so I wanted to know, does that affect the quality of the DNA target that we're essentially isolating? And when I compared it to Trizol extracted genomic DNA, we found that the biosensor was able to detect both, and the level of detection was still similar. So that was really nice too. So at this point, now I've eliminated PCR, and now we've also eliminated the use of another expensive reagent. So we're bringing down the cost at every single step. So again, right, this is genomic DNA. So the main question is, does this really work in real-life application? So we wanted to know, first off, right, if we're thinking about bioterrorism threats, what if someone spikes the water, or what if they spike a dairy, right? Or what if they spike another type of manufacturer of a food substance that's in a liquid? So we wanted to test, will it work in finding our salmonella within a liquid food matrix? So in order to do this work, we essentially just spiked either whole percent, like whole milk, or 100% orange juice. The reason we chose both of these is because they're two different liquid environments. One is basic, one is acidic, right? So that can affect the target, right, which again is DNA. The other thing about this is that previously we were doing it in pure culture, which is bacteria essentially grown in nutrient water. So it's a clear, essentially liquid. But here we have different consistencies of liquid. Not only are their pHs different, but their viscosity is also a little different as well, and they're opaque. So doing this work, again, we spiked milk and or orange juice. We just took a 1 milliliter sample, or for some of you, if you're in the medical field, that would be 1 cc. And we isolated DNA from that using Trizol, right, because it was an hour and a half, and we applied it to the biosensor detection system. And so to begin, when we have pure bacterial culture that is, so just salmonella spiked into the milk, the way we did this is we grew salmonella overnight, we added it to some milk, let it stand at room temperature for 20 minutes, and then we just took a 1 milliliter sample before isolating the DNA. And then we applied that to the biosensor, and we found that we indeed were able to recognize the target using the biosensor. The other thing that we also tried was that in reality, we know that there's going to be more than one bacteria in that system. It's not just going to be salmonella. And so we wanted to kind of recreate an environment where that milk isn't just with salmonella. We also introduced E. coli, and we also introduced Shigella. So 3 different bacteria, one being our target, the other 2 not, to see if, again, it would be specific. And so that would be the mixed bacterial culture. And so when we did that, we saw that once again, at the 100 nanograms per milliliter of DNA target, our biosensor was able to work very effectively. Once again, seeing that nice hook effect, right? The more target you have, the more messy things get. The other thing is that, again, this was in Trizol, but for proof of concept, will this also work using the standard phenylchloroform method? And we were able to see the same. And if anything, we slightly got better results. Okay? So these DNA extraction methods, I do really want to point out that it's just basically quick and dirty. There's no purification of the DNA. There's no amplification of the DNA. You basically get your sample, isolate the DNA, and move on. Okay? So when we look at orange juice, we see once again that our system, our biosensor, is able to detect all the way down to 10 to the 4 colony-forming units, right? If we think about how much bacteria we actually spiked with, as well as just the DNA target itself, right? Here's the 100 nanograms per milliliter. So the next step then, so the very first runs that we did, those were essentially proof of concept. And of course, it's a liquid where things are nicely diffusing. But the reality is, if we're going to really apply this for the food supply, if you will, right, not everything's a liquid, things are also solids, and the more solid they become, the more viscous that sample can possibly become, impacting your ability to essentially isolate that DNA. So the next step is to really validate it within a solid food matrix. And so in order to do this work, we looked at 4 different common, right, problems: eggs, raw chicken, dog biscuits, we know they get recalled all the time, and then of course spinach, right? So all of these have been at one point or another recalled for salmonella contamination, and so those were our test agents, if you will. So very similar scheme again, grow up that salmonella, spike the food, extract the genomic DNA, and try to sense it with the biosensor. And before we begun, we first wanted to really reproduce this, right? So the various examples that I showed you, those were essentially 1, 2 trials, maybe 3 trials. So we really wanted to put it to the test. So here we wanted to really establish reproducibility more than the standard 3 times. And so here we did this 7 times. This is just with genomic DNA, but it's pure. And so there's no spiking of food or anything like that. But we could see that just straight up genomic DNA from bacterial cultures are good. The one thing that was really nice about this is that we were able to get it all the way down to essentially 63 nanograms per milliliter. So we were able to kind of bring down that detection level even more so. So our first, I guess, culprit is raw chicken. And so when we spiked raw chicken, and we spiked with different things. So let me just orient you. So we spiked with just salmonella culture by itself, a mixed bacterial culture that also contains salmonella. So for these, it was salmonella, bacillus, and E. coli. It was a mixture. Over here where it says nonspecific is essentially just the E. coli and bacillus. So there's no salmonella here. And then over here, this is essentially just water. So there was no bacteria spiking whatsoever. It was just water. And what we found was that, right, just so here where we have salmonella by itself and E. coli by itself, that is essentially just DNA target. And so that's kind of where we set our limit. And what we found is that, once again, the biosensor was doing a pretty good job at being able to detect our DNA target, right? So whether that was it being spiked with salmonella alone, whether that salmonella was in a mixed bacterial culture, right? And when we look at no bacteria whatsoever, we see very little signal that's not statistically significant. But the one thing that was really standing out to us is that non-specific, right? So again, not salmonella. These are those that are spiked with E. coli and bacillus. That was also giving us a signal. But that signal was sometimes interfering. So depending on which one you looked at, right? Sometimes it wasn't significant, but sometimes it was. So we were definitely still having an issue with background, right? So there's still more work to be done. When we look at spiking spinach, we see once again that the biosensor is being more effective at being able to pull out that target. But when we looked at dog biscuits, there was a lot of variation. That was a no-go. This was not working out at all. And when we looked at egg yolks, it was the same thing. So what we learned from this is that the one rate-limiting step here is the consistency of the sample that you're isolating that DNA from. The more watery it was, right, the more dispersed it was, the better the biosensor was able to detect the target. But if your sample that you were isolating that DNA from was more viscous, as what we saw when trying to get a sample from the dog biscuits that are dry that we have to apply water to, right, or the raw egg that itself, we already know that egg yolks are very, very viscous, okay? That was interfering with the whole extraction part. So that's the rate-limiting step here. So putting it all together with the validation of this particular biosensor works really good in liquid food matrices, right? Because, again, the consistency of that sample is nice and loose. But when we move it to a solid food matrix, right, it gets a little tricky because it wasn't very effective in that egg yolk or the dog biscuit. And because we saw a lot of high variation within those samples, and we're pretty confident that it's because of the ability to be able to extract out the DNA. So future efforts then, as we think about validating biosensors, right, is not only do you have to put them to the test, knowing that your analyte isn't just going to be in a vacuum, right? It's just going to be out there hanging to be able to be recognized, but it's going to be in a mixture of things. You really need to focus on reducing that background noise, improving that sample preparation in a manner that is going to be easy and fast, and then also reducing the time to detection. So right now, when I first began to use this biosensor and the way that they had set it up, the whole process took about 8 hours long. That's not a very good system when you want to commercialize something, right? You want to bring that down to just a matter of minutes, maybe even possibly no more than 4 hours, I would say, because it's essentially halting the production line. So reducing the time to detection is also really important. And then, of course, once all that gets worked out, we want to be able to recreate this targeting other targets. So how do we do this, thinking about this process of streamlining the actual detection process? So again, there are essentially 4 main steps using this particular DNA-based biosensor, right? When we first began, isolating the DNA using Trizol takes an hour and a half, right? If you do the phenylchloroform method, it actually takes 2 hours and a half because you have to heat it up and you have to do all this stuff. But if you use a commercially available kit to isolate that DNA, it takes 30 minutes. So the studies that I've been doing now are using a commercially available kit, so it takes 30 minutes to get your genomic DNA. The hybridizing to the magnetic nanoparticles, right? They were erring on the side of caution, so they were hybridizing them to, an hour and a half, 2 hours. I've been able to reduce that down to 60 minutes. Same thing with the gold nanoparticles. I've been able to reduce that down to 60 to 90 minutes. There seems to be no difference between that. And then once you actually apply it to take the voltammetry, that just takes 7 minutes. You can't get away from that at this point. So reducing it down is really important because when we compare it to what other agencies are using right now, so again, these are things that are either on the market or have been published. Detection times, right? We got about 9 hours, right? 9, 10 hours, 7, 8 hours. That's a pretty long time. This is taking about 4 hours right now, 4 hours to detection time. The cost, right, of doing it with the conventional way could be anywhere from $10 to $40 per sample. Doing it in this way, it costs about $2 per sample, right? So this is really, really important. Cutting out very expensive steps, cutting out the use of high equipment, right? So equipment and instrumentation that is costly to maintain, costly to get. That's all really important. And again, the various reagents that you use in these processes are also really important. And so reducing that cost is also of interest. So here's just a little bit of a reference slide. And I want to thank the 2 funding agencies that supported this work, my collaborator, Banji Elohia at Michigan State, and then the 2 undergrads that have worked on this project. Jessica is currently at UCLA getting her PhD in biochemistry, and Mason is currently at the University of Oregon getting his veterinary science degree. So that's it. Thank you so much for your time. And I'll take your questions. Okay, so your question is, what is the cost of applying, right, essentially functionalizing those gold nanoparticles with the nucleic acid primers, and also what's the shelf life, right? How long can we actually keep those for? So interestingly, when I first took this project on, the postdoc that was working on it, he would spend so in order to functionalize those gold nanoparticles, it takes 3 days, right, before you can even use them. And so he'd spend 3 days functionalizing them and then do 1 run and then have to start all over again. And so that's not very efficient. And so I essentially asked, well, how stable are these? And this is something that the lab had already worked out. And they had found that if you functionalize them, they're good for about 30 days at just like in a refrigerator. Right? So you can actually functionalize a lot of nanoparticles for your target and then store them for about essentially a month, which is really good for the nanoparticles. Now, the other thing about that is that DNA can be, we know that it's pretty stable at room temperature too, but the reality is that the outside environment isn't room temperature, right? It's going to fluctuate a lot. So there still is the need to stabilize them roughly between, you know, I would say 4 to 8 degrees, and that just requires the use of a small refrigerator. So the thing about that specifically that I want to mention is that the potentiostat that needs to do the voltammetry, there's handheld ones, and so those have been tested in the lab, and those you can actually take into the field. You can also have small incubators, because we have some of those, that essentially run off of a car battery. So the refrigeration, I guess, that you need for your reagents and the power supply that you need to run your instrumentation is no more than a car battery. So this is very field applicable. As far as the cost of the actual primers, so recently I put in an order. I was able to make enough for about 50 runs, and the cost of those primers was, I believe it was like $100, right? And these are primers not just for the gold nanoparticles, but also for the magnetic nanoparticles. So that was enough for both. And the thing about those primers is that the more you buy, the lower the cost is of the primers too. The most expensive thing, interestingly, in all of the reagents in this are the screen-printed carbon electrodes. That's the most expensive piece in this whole thing, which was really interesting to me when I first began. Other questions? Mm-hmm. Okay, so thank you for asking that. The question was, how much training does the individual need to have in order to be able to do the biosensor detection and use that system? And so I recently, actually just this summer, trained high school girls, so these girls were sophomores and juniors, to use this system. So, you know, they have a high school level, so they were 10th graders and 11th graders, right? So their age is, I want to say 15 to 17 maybe at the high end. And they were able to not only understand how it works, but to be able to run the whole system. They were able to learn how to isolate the DNA, hybridize it to the magnetic nanoparticles, do the wash steps, to the gold, do the wash steps, and actually do the voltammetry and understand how that works too. So I would say high school level of education at minimum was needed. Obviously, I've trained college students to do this, and so when we think about level of education, at minimum high school. Thank you very much.